Cisco genome sequencing project. This is a work in progress, so it's somewhat preliminary. But I'm also going to take some time to present a little bit of a synthesis of of research that's been happening in my laboratory, along with collaborators over the last couple of years. And try to talk about how all these projects might fit together, hopefully somehow in a coherent fashion. But I thought I'd start by talking about the theme of this conference. The organizers did a really wonderful job of choosing what I think is a really important topic of coldwater fisheries in light of climate change. So as I'm going through this presentation today, I'll be thinking about, try to ask you to think about how this might fit into this overall theme. I'll come back to that at the end. The other thing I want to mention before I jump in is my co-author, Nathan Backenstose, is a PhD student in my laboratory at the University of Buffalo. And in particular for the genome assembly side of this, Nate's done really all of the actual work on this, so I just wanted to acknowledge him at the start. So this work is motivated by a quest to try to understand morphological diversity in Cisco. We've been thinking about this for a number of years. In particular, in places like Lake Superior, where there's a lot of Coregonus diversity still in existence. If you go out, and how many of you have tried to identify larval Coregonus? Yeah, exactly. It's very difficult, right? So different species might look very similar. They're hard to identify. But over development, once you become adults, we see much greater differences among morphological forms with the adults. So for example, an adult kai looks very different from an adult artedi. So we're really interested in this development and how these differences come to be. Morphological differences are found in large part in the head, differences in eye diameter, for example, jaw openings, for example, as well as gill raker structures, size and number of gill rakers, all of which ultimately which plays some role in not only the morphology, but ultimately translates into ecological variation in nature. So what causes these this morphological variation? Well, we know that to some extent it's just simply a reflection of their environment that they are raised in. So phenotypic plasticity shapes quite literally some of the morphological variation in Cisco's. We also know from a variety of laboratory experiments that there are in fact heritable genetic differences among forms or species. And this is what we really want to understand. What are the genetic markers that underlie this functional diversity in Great Lakes Coregonus? And forgive me for the Lake Superior focus here. I hope that some of this will apply to, say, Lake Ontario as well. So I mentioned a number of morphological characteristics or morphological traits that vary among these species. We're also thinking about how those translate perhaps into ecological variation, things like life history variation, but also aspects of where these fish prefer to be, what kind of habitats, depth preferences, and so on. I'm not a fisheries biologist in the strict sense, but I've heard these terms mentioned, and I know these are really important to those of you managing fisheries. And ultimately, this is what we're thinking about, is how do we connect morphological and ecological variation to the really important questions about fisheries and fisheries management. Okay? So what we're trying to do is bridge across these different data sets. So our objective in this research is to determine not only how Great Lakes Cisco's differ across these various data sets, but also why they differ. So mechanistically, what's happening in their biology that produces this variation. We're taking a genetic focus here, but trying to pair it with different types of data sets. So what are the functional genetic changes that cause this variation? That's our key question. As I said, we've been looking in Lake Superior at some of the extant diversity there. We've done a variety of studies trying to link genetic variation with morphological, so body shape variation, as well as things like stable isotopes, which might reflect diet preferences. And what you can see in these principal component analyses, and for isotopes, going two variables, carbon and nitrogen, stable isotopes, is that patterns of variation among these 4 different forms are more or less consistent across data sets. That is, genetics to some extent can predict morphology and so on, and as well as vice versa. So we think that this is potentially a fruitful approach for trying to understand the linkages mechanistically between these data sets. The question is, when we see these patterns, what we as geneticists really want to understand is what are the actual genes, the specific parts of the genome that are important for shaping this variation. How do we make these connections? So I'll talk about kind of an approach that we use to try to start with all the genetic diversity that's in a genome. So we start with all genes. We're having this sort of dartboard kind of approach that we're taking. We start by looking at all of the genes in the genome, and then we have a variety of statistical quantitative methods to try to identify which are the really important parts. So we try to filter out genes that might not necessarily be important for morphological or ecological variation. And what we end up with is, in the center are these sort of bullseye genes. In this case, we identified 21 of these genes that look to be really important with respect to morphological or ecological variation in Cisco's. So we've winnowed it down. We start with all genes. We filter out those that we don't think are as important. And then we identify these sort of bullseye genes that we want to focus on. Once we've done this, these candidate genes, as we call them, are then put under a little bit more scrutiny. We want to really understand what's going on with these genes. So I'll give you one example of where we've done that. We followed up one of these genes called rhodopsin, which is an important vision gene. And I'll just give a quick example of how that, how their functional variation might relate to ecological variation in this gene. So these are data, actual data from our samples that we've used in our Lake Superior study. We have 3 shallow water, relatively shallow water. I realize these are there's a lot of variation, but so artedi, foyi, or floater, and shortjaw were collected in this case in relatively shallow water. We have a deeper water kai here. We've looked at DNA sequence level variation in rhodopsin, and the pie charts reflect differences in allele frequencies in those genes. So as you can see, kai is completely different, or nearly completely different from the other species. So what does rhodopsin do? This is a protein that's expressed in eye, specifically in the retina, and it's a gene involved in that controls low light vision. So down in the deep dark waters of, say, Lake Superior, we think that this gene may be responsible for some of the functional tuning of how deep water low light vision functions. The exciting result of this is we've identified 3 changes in this protein that actually affect the amino acid structure of the protein itself. One of these changes, the one that's the most different among these different species, is the exact same amino acid change that completely independently in some deep sea fishes underlies adaptation to deep water environments in marine systems. So totally independently evolved systems. And that makes me kind of tend to believe these results more, that it's the exact same change that happens in this protein. So we think this gene rhodopsin is really interesting in light of depth preferences in Cisco's, how they can tune their genetic background to matching particular environmental conditions. I'll just put a plug. We have a student, an undergraduate student, Katie Eaton, has a poster on this with some more details if you're interested. That poster's here. We've developed these specific genes that we think are important. We're developing these assays to be rapid and flexible, so we can sort of genotype these individuals quickly. So this is new long-read DNA sequencing technology, something we're really excited about, in large part because of the portable nature of these sequencers. So this is a small thumb drive-sized DNA sequencer, and we can do things like take it out into the field. So the idea is that we want to make rapid, easy assays that people, including students, can go out into the field and genotype Cisco's or other species. So we've identified some of these genes that we think are important, but we keep going back to this problem of how to not not knowing the full context in which these genes are interacting with one another. So we don't know where these genes are in the genome because nobody sequenced a Cisco genome just yet, or any of the close relatives. So that sort of motivated this idea that we wanted to go in and try to sequence the Cisco genome. I'm presenting here an extremely simplified overview of what that process is. I'm happy to talk about the nitty-gritty details if anyone's interested. This is work, again, by Nate Backenstose in the upper right photo. We're using this long-read DNA sequencing technology again. And so I'll just emphasize it's a work in progress, but we wanted to start getting the community talking about this assembly because we want it to be made available really early on and of use to other people. So we're trying to get this out as quickly as possible. So just the raw sequence data with this long-read DNA sequence technology, I just want to make one point here, is that we're able to generate very long fragments of DNA sequence with this technology. So this histogram shown here corresponds to individual read length of single DNA sequences. So the peak is about 47 kilobases, so 47,000 bases long. These are individual DNA sequence fragments. And that's much longer than, say, standard DNA sequencing, which is about 150 base pieces. The reason why that's important, if you're trying to put together a really big complex puzzle, having large pieces makes it much easier to do. So this is what we're striving to do. Okay, so some details on the Cisco Genome Assembly Project, where we're currently at. We've done several versions of this over the last couple months. Our current version, we're calling 1.0, is shown here. Some of the details. The important point is that the total length of the assembly, so how long of DNA sequences have we assembled, the total length is about 2.5 billion base pairs. That's exactly approximately what we would expect based on the genome size. So that's good. That means we have a mostly complete assembly. How good is the assembly? There's a variety of metrics for this, but the last column is one particular assembly. This is how big are the individual pieces that we've assembled. So 458,000 base pairs on average. What does that mean? Just for perspective, another existing genome project that's now publicly available, this is European whitefish, we're sort of on par with that level of completeness for contiguity with the genome. So we're making progress. We have a long ways to go, but we're on our way there. I wanted to just take one more minute and show you what an assembly actually looks like, some of the ugliness of a genome assembly that's in the works. This is a graph level assembly. Individual lines reflect assembled pieces of DNA. And you can see there are many pieces still of DNA. Ultimately, we want to get these pieces assembled into chromosomes. But we made good progress there. But we still have some work to do to stitch these individual pieces together. You'll notice in the upper left this sort of hairball, all of these complex pieces stuck together. That's because salmon genomes are very complicated in structure. They have a lot of DNA sequence repeats. This is a huge challenge for assembling genomes, but I want to show you this sort of ugliness of what we have to deal with and why it's taken so long to get to a proper assembly. I mentioned the complexity for two reasons. One, to show you that we have a lot of work left to do, but number two, because if we think about how much time we spend trying to understand Coregonus or Cisco biology, relatively little effort has been given to trying to understand this complex genome that these organisms have. And I think if we think about potential for understanding, making new biological insight, going in and looking at this complexity, it presents some opportunity because we haven't dug into these kind of complex areas in the genome. With that, I'll just circle back to this theme of the meeting, cold water fisheries in light of climate change. You may be asking, why do we care? Why do we need a Cisco genome assembly? Very quickly, I'll just say that this allows us to integrate all genetic markers that have happened to date. So providing this assembly could provide a common reference where we could take microsatellites or other genetic loci put them into the same genome assembly and try to see which ones are close together, and so on. And this allows us to try to understand why they may give conflicting or not patterns in various studies. Refining genetic marker panels, an ongoing effort. People like Wendy Stott and Wes Larson's lab have been making great progress here. I think this could contribute. Linking genetic and morphological variation to fisheries variability. And then finally, thinking about, in the context of climate change, what are the sources of resiliency or not in Coregonus, in Cisco's? And we're thinking about this idea of a genetic portfolio effect, how diverse genetic background may or may not shape their response to climate change. So we have to go in and dig into this hairball, try to untangle this snarl and make sense of it. That's an important goal. I've presented snippets of many different projects. We have a number of collaborators, and I just want to acknowledge them, as well as our funding agencies and some folks who provided samples and various assistance.